Okay, let's continue. Someone asked about how to find the score in the page 6. Let me first answer that, then we will continue with our lecture. So, about this slide, I think you asked. For the rest of the sequence, if you want to find the rest of the scores, and in that case, we said take a look at the frequent use matrix. But the thing is that we didn't give you the values for match and this match or mismatch because it's related to this specific metrics. You don't have the value manually. This one, for example, if you use a software or you use a database related to that, then you can come up with the answer. Otherwise, manually, you can't come up with the answer like this. Okay, so this one just was an example to show you that what are our possibilities and approach. That's it. But you cannot find the total score based on what I've given you here. Okay, for optimal alignment, this table that we find about it by using different approaches. For example, you can see the name of the guys who came up with the algorithm. For global alignment, we usually have this approach. For dynamic programming and for local one, we have this one. Now, this slide is very important in order to see how we can find out a possible alignment by just looking at these metrics. Of course, you cannot find the best one again through it easily. And this is more complicated than what it looks. There are different possibilities. But again, you have eliminated many possibilities already. And that's the beauty of dynamic programming, because it makes your job much easier. Because if you consider all the combinations together, then you would need lots of time and space in order to deal with that. But when you have just a polynomial solution instead of exponential one, so of course this can make our job much easier. Now, take a look at this table to see what we can do, for example, to find the best alignment as much as possible, or better to say the options that we can have. So start from the final number that you will find in the table. As I said, sometimes they call this cell as the optimal value that you will find. But in fact, it's better to call it initial value, because you start from the initial value and you trace back to the first cell. So how we can do that? Which path can we take? So take a look at its neighbors. We calculated this one by using these neighbors anyway, right? So take a look at those values and choose the maximum one. But is it always the maximum one? No, because it's more complicated than that. But usually we prefer the maximum. Now, if I see the same numbers, I usually prefer to go in a diagonal format, because now I align a letter with another one. As soon as you go up or you go to the left side, it means that you are considering a new gap. And as you know, a new gap can have a high penalty. You usually try to avoid it as much as possible. But sometimes sacrificing one cell can give you much better score at the end. So we go forward diagonally as much as possible. So 1 to 0, then 0. Again, I want to move on and I see -1 is bigger than -2. Then I choose the diagonal trend. Again, next one. And this one, you see, -1 is bigger than -2, but we didn't choose this one. It was even possible you choose this one. This is one option, okay? I would, for example, to make my job easier to run my simulation in order to define my algorithm, maybe even before simulation, I prefer to choose maximum so that when I'm typing the program, it's much easier for me. Otherwise, how a computer can decide about these things? Just by using heuristic method, and we will also talk about heuristic method in the next session, that what I'm talking about related to that. So you reach, for example, this one. Why we chose this one over these, there is no explanation that why they prefer this one. As I said, it's also a good idea to move on in diagonal trend. But you see, if I keep doing it, I will reach finally -3. I want to avoid it because at the end I should reach this one. So where is the best place to consider gap? And this is also one important question. If it was me, I would prefer to use this path. For example, when I reach this one, choose maximum. This one again is maximum. This one maximum, again maximum. And as soon as I see conflicts, I prefer to choose the diagonal one. You see, I think this one could be much better way to deal with this alignment, okay? But they have shown these two options, and I don't know what was their reasons, but I believe this one can give you much better identity. I mean, the higher percentage for identical nucleotides, and even if you have mismatch or gap penalties, it will give you better score at the end. Now let's take a look at the second matrix. In this one, we have two optimum answer, because as I mentioned before, they sometimes call it optimal score, which I don't like, but we can say we have two initial points so that we trace back to the first cell. But in local alignment, as you can see, for the first trend, you don't stop at the first cell. You stop somewhere even in the middle of the table, you see? And for local alignment, that where should we stop? This is also very challenging then. What's the point that we should stop and again jump to the next optimum one? You see, it's a big question and it's not easy to answer that. But why local alignments are important and why they are useful? Because sometimes if you just notice about the regions inside your DNA or protein, it can solve many answers. Like if you find a similarity, for example, more than 80% or even exactly 100%, maybe you can find homologous, like the ones that we mentioned before. Do you remember about the name of them? That, for example, you had homolog genes, or you had different part of them, like paralog, ortholog. You can find them by using local alignment as well sometimes. But I also mentioned about global alignments and how they are useful for finding homologous genes as well. Okay, but even for these ones, especially when we talk about domains of protein, that one can be very useful, okay? Because usually for homologous genes, because we want to compare two genes together, all of the sequences, global alignments could be useful. But even don't just eliminate this chance that you can use local alignment in order to even find homologous genes. It may sound not right, but this is possible. So by taking just looking at these two tables and comparing them, you can understand that what are the differences between local and global alignments better. Of course, we will go through the details, and that's the place that we will introduce you officially local sequence alignment. We have a term named terminal gap. When we say terminal gap, is a kind of gap which happens, or we apply actually, because in nature we don't have such a case. We just use them in bioinformatics. So when you consider gaps before and after your sequence like this, not in the middle of your nucleotides or amino acids, in that case we call it terminal gaps. But why they are useful? Because if you are comparing two sequences together and one of them is very long and other one relatively is much shorter than the other one. In that case, terminal gaps could be very useful for us, because assume that you have this sequence, and this one 100% matches this sequence inside the other long sequence. So the rest of it you can consider terminal gaps, and only this part, which is the whole sequence, is exactly the same as this one. So such alignments can be very useful for us, and there are some methods in biology that they can splice, for example, this sequence, for example, from a mouse, and just insert it in one other cell and cure something. And based on that, they understand if they do the same for human beings, maybe that could be the answer. And by the way, just cutting one part of the sequence in molecular biology is not as easy as it sounds. It has its rules. But as computer scientists, we don't care about that part, because we're just talking about the theory related to molecular biology. And this is their job in order to practice it in nature. But of course, it's not a good excuse to say I don't care about molecular biology and I just consider letters and that's it. This is my job. But this is not a good idea either, because you should know the background related to molecular biology. You don't want to just simulate something which is not real, you know. But some part of it, yes, this is not our job anyway. For example, what's exactly happening in the labs, the details. Okay, so for example, the difficulty, we don't care about the difficulties, you know. We just simulate and say, okay, this is the best way. If molecular biologists can do it, perfect. And if this is expensive, okay, this is not our problem as computer scientists. Of course, when you are in big projects and you are working together, and sometimes molecular biologists in the group may force you, for example, to eliminate one possibility and move on to the next, and this is totally normal. And in that scenario, we may sacrifice our solution for that problem. Anyway, now this is a good question. That should this alignment give us always 100% accuracy? And can we waiver penalties for terminal gaps? It means that, you know, if I consider penalty for each one of these gaps, it will be a huge negative penalty for us. So yes, it is possible sometimes to even neglect terminal gaps as penalties. This is possible. And shall it be always 100% the same? No. The answer is no. For example, if here you have, instead of G, C, then you will have a mismatch here. It doesn't matter. Sometimes even you reach 80% similarity, that's good enough for us. It depends on the case, and sometimes even having a specific codon and you can match it with another one, it will be great. So not always it should be 100% the same. So here you can finally get introduced to local sequence alignment officially. We had a slide before. Related to global sequence alignment. But here, let's see, what do we mean by local sequence alignment exactly? So you assume that we have two strings, S1 and S2, and the goal, let me finish this one, then I will check your message for sure. So the goal is to find substrings of S1 and S2, which their global alignment is optimal. This is very important. It means that you are dealing with just one substring, not the whole sequence. And then when you choose the substrings, then you compare these two substrings from S1 to S2 as we did in global alignment. So the algorithm is exactly the same. As soon as we find out about those substrings, and that's the challenge for us in local sequence alignment. For example, like this one. Now we consider these substrings together. From S1, let's call it S1 prime and S2 prime. So this one is S1 prime, and this one is S2 prime. If you compare them together, 100%, we can see the alignment. As I answered this question in the previous slide, should it be always 100%? Not necessarily. If it was A, GG instead of just GGG, then in that case, still maybe it could be a good local sequence alignment. So I will go through the way we apply dynamic programming for local sequence alignment in the next session. And I will also talk about heuristic methods that we can use in bioinformatics briefly. So after 5 minutes, I will explain about first assignment a little. I can't give you the solution because, as you know, you have a deadline until Friday, and you will get feedback, for example, if you submit it, and personally I give you feedback in the assignment link. But let me give you some hints anyway, just after 5 minutes break.